our yet another episode of the COVID-19 conversation series presented by NepSciHub. Our last episode with Dr. Daniel Griffin was attended and appreciated by many people, and we sincerely hope that you'll find this critical conversation equally relevant and insightful. My name is Jolia Mathia. I'm from Nepal, and it is my pleasure and honor to introduce you to our special guest for today, Dr. Monica Gandhi. She is the Professor of Medicine and Associate Division Chief of the Division of HIV, Infectious Diseases, and Global Medicine at the University of California at San Francisco, UCSF. She also serves as the director of the UCSF Center for AIDS Research and also as the medical director of the HIV clinic at San Francisco General Hospital. Dr. Gandhi has been a tremendous source of knowledge for clinicians and policymakers since the pandemic began. From being one of the first scientists in the USA to recommend universal masking to advocating for global vaccine equity, she has not been one to be afraid of fighting for what she truly believes in. With such a distinguished background in infectious diseases and public health, she's the ideal person to guide us in controlling this deadly wave that is rampaging through Nepal and India. She will be discussing about vaccines, variants including B1617, the Indian variant, this current catastrophic surge, and public health strategies that we can implement in the short term and long term to mitigate the damage. She's also been a fierce voice for global vaccine equity, which I'm very, very interested to hear more about. Like last time, the format for today's webinar will be a presentation by Dr. Gandhi, followed by a Q&A from our audience. I would like to give a heads up to all our audience to use our chat box on your screen. We'll try to address as many questions as possible. So without further ado, I'd like to quickly ask Dr. Sargam Mahat, our moderator for tonight. He'll be facilitating the conversation and the Q&A. So the floor is all yours, Dr. Mahat. And thank you so much, Dr. Monica Gandhi, for your valuable and precious time. We really, really appreciate it. I'm really looking forward to this discussion for tonight. And I also have some questions for you as well. Thank you so much. Dr. Mahat.
Namaste and good evening, everyone. So first, before we start our presentation, I just want to say something about this new initiative that we created. It's called NepSciHub. So we created this YouTube channel, this initiative with the sole purpose of disseminating evidence-based and data-driven information to our Nepalese community. As we all have seen, COVID-19 has led to infodemic of some sorts, and we are concerned about the amount of misinformation being published by mainstream media. We aspire to be a hub where people can find evidence-based information devoid of any political bias. With that said, we are extremely grateful to Dr. Monica Gandhi for agreeing to do this today with us. And I personally have learned a lot from her publications, online appearances, her tweets. I followed her for a long time and I really look up to her. So it's a huge honor for us to have an expert like her amongst us to share her knowledge on vaccines and variants and others. So it also comes at a very relevant time because Public Health of England, PHE, just designated B1617.2, which is a sublineage of B1617, as a variant of concern today. So without further ado, I would like to hand this over to Dr. Monica Gandhi. Thank you so much.
Okay, thank you very much for having me. And I will share my screen, and then just so we have time for questions, I will try to go through this relatively quickly. We'll be talking about strategies to protect oneself outside of vaccines, but then really concentrate based on the questions on the vaccines. And so, when we talk about the coronavirus, one thing I want to mention is this is not the first time we've seen a coronavirus causing a terrible, severe infection. And this is the first time, however, that we developed a vaccine for such, because the other two coronavirus pandemics, specifically SARS that went from 2002 to 2003, about only 8,100 cases, 9 months in the world. This particular pandemic did not require a vaccine because it was syndromic surveillance. It was symptom-based management, and it went away. And essentially, the Middle East respiratory syndrome, same thing. It was a very mild, it did cause severe disease, with the coronavirus, but it only lasted, it lasted a shorter period of time, but importantly, it really was linked to mild disease. It was linked to severe disease, but it was really very limited because it was based on symptoms. But what about this new coronavirus? The reason we needed a vaccine so desperately is no matter what happened, this is a completely different virus. This is a virus that we identified as a new coronavirus on January 7th, 2020. On January 30th, it was declared a global health emergency. On March 11th, it was a pandemic. On March 26th, the US became the epicenter of the pandemic. And then with vaccinations that receded, and March 5th, 2021 is when cases, you started seeing the rise in India. And now we're in our situation that we have today in India with spillover of obviously this effect into Nepal. This is truly the great viral pandemic of our day. And this is yesterday, the cases around the world with all this increased incidence, obviously in South Asia. And at this point, there have been 3.26 million deaths since the beginning of the pandemic. And we've had the solution to this pandemic actually since November 9th, which is when the first press release came out of the first effective vaccine, which is why we will be talking about global vaccine equity and how we could have achieved this. We could have worked, the world could have worked harder on this for India. This is the latest coronavirus map and case count that you all very well know. Cases right now in India have been increasing really since that first bit of March and very exponentially. And deaths at this point are at 234,000, unfortunately, and the 14-day change keeps on going up. And Nepal is seeing, this is from May 2nd, but seeing this increase in obviously the spillover from India's terrible outbreak of coronavirus. The three non-pharmaceutical interventions for COVID-19 have changed a little bit. I mean, there has been questions about outside and all of this going on very actively, even today, in the CDC recommendations. But essentially, it still stands that the three major mitigation procedures for COVID-19, while we're waiting for the most effective solution, which is vaccination, are masks, ventilation, and distancing. And the ventilation is actually increasingly seems to be becoming the most important part of this, meaning outside is so much more safer than inside because viral particles disperse very easily in the outside. And indoors does require masks, and it doesn't just require masks, but good masks. And I want to talk about a little bit about fit and filtration of masks before we get to vaccines. The fit and filtration aspect of being inside means that you want a tight-fitting mask that either is fitted to your face so that it's not gapping on the sides, or if preferable, or if possible, a surgical mask with a cloth mask. Why? Because what a surgical and a cloth mask does is the surgical mask blocks viral particles from sort of electrostatic repulsion, and the cloth mask blocks viral particles through the physical cloth. And so putting those two together has been shown to improve fit and filtration to an equivalent level of an N95. And the reason that's so important is right now in India, and giving India and Nepal, given what's going on, I would absolutely recommend stronger masks for inside. The CDC also looked at this and recommended either, again, a cloth or a surgical mask together, or looping the surgical mask over your ears so that it's very tightly fit across the face and tucking in the sides. They call this tuck and fit, but that's also important. So you want a very fit and filtered mask. And really, it can be different for outside and inside. Outside, the ventilation has you so protected that outside you can wear, you don't have to wear one unless you're in a crowd. Right now, being in India is just being in a crowd, meaning given this much coronavirus, I would absolutely still wear a mask outside in India, especially if there's closed spaces, not if you're alone. But it really is a simple mask because the ventilation has really taking care of the virus for you. It's really inside where we need these more fit and filtered masks. Outside transmission is true, and this gets into this aerosol droplet debate, which I'm not going to get into right now. But essentially, it's just true that any large epidemiologic study has shown that outdoor masking is much more outdoor transmission is much lower than inside transmission, maybe about 5,000 to 7,000 times lower. So this is a safer place, is to be outside. Okay, because I want to get to questions, let's get into these vaccines. So, though the top 3 are the ones that are authorized in the United States, we will spend time on the Astrazeneca, which is called Covishield in India, and the Bharat Covaxin vaccine, and know that they're different types of vaccines. Really, there's a division between spike protein vaccines and whole inactivated virion vaccines, of which Covaxin is an example. So and by the way, just when I go through these, just to be fair, we don't have a peer-reviewed publication on the Covaxin product. We have an updated press release from April 21st, but we don't have a full peer-reviewed publication. We don't have that for Johnson & Johnson either. So it makes it harder to give as many details about those products. But AstraZeneca, we have quite a bit of information on. Most of the vaccines, except for the Covaxin, the Sinovac, and the Sinopharm, involve the spike protein. So Covishield, AstraZeneca involves the spike protein. And what is a spike protein? That is really the protein that binds the virus to the host cell. That spike protein sticks out of the virus, binds to the host cell, and it's really what these vaccines are doing, are coding for the spike protein and the receptor binding domain. Or they actually give you the spike protein in the case of Novavax. So to be very specific, the 3 types of vaccines that involve the spike protein are the mRNA vaccines, which the only 2 examples are made by Pfizer and Moderna. The adenoviral vector DNA vaccines. The mRNA vaccine, just to give a little more understanding of that, is it's literally a piece of mRNA that makes your body code for the spike protein. It's put in a lipid kind of nanoparticle and injected into the body, and then the mRNA molecule comes out, and then you actually translate that into the spike protein. The adenovirus DNA vector vaccines, the 3 examples are the Sputnik 5, the AstraZeneca, which is Covishield, and the Johnson & Johnson. And those vaccines involve a very benign virus called the adenovirus that doesn't actually replicate in the body in this particular way, though they cause cold symptoms in if it was given de novo. But it's a adenovirus with the DNA that codes for the spike protein inside of it. And then again, your body makes that into mRNA, into protein, and then you raise an immune response against it. And then the inactivated whole virion vaccines are the part of the product, the Covaxin, Sinovac, and Sinopharm. So these are the differences in how these different vaccines are made. And when we talk about the vaccines and when we talk about their efficacy against the B.1.617 or any other variant, it is really important to remember how the immune system works. And it's quite simple to say that, not simple, but it's quite important to say that antibodies are one arm of your immune system. They come from B cells. But actually, the arm of the immune system that's most enduring against viruses are T cell responses. And T cells are divided into CD4 cells, which are called helper cells, and into CD8 cells, which are called killer cells or cytotoxic cells. And what you want your vaccine to produce is a strong T cell response, specifically what's called Th1 CD4 response. That's the CD4 response that's geared towards fighting viruses. And the important thing about all of these vaccines, except for a couple, is that they actually took the trouble, including the Covaxin, to measure and quantitate T cell responses to the vaccine along with the antibody responses. And the reason that's so important is when we talk about B1617, it is important to remember that these vaccines in their trial design took the trouble to measure T cells and showed us that we mount strong T cell responses to these vaccines. T cell responses are very important because fundamentally they modulate the severity of disease, so that even if you get an infection, it is bound to be, if you have a good T cell response, it's bound to be either asymptomatic or mild. So even these breakthrough infections that we've seen in the context of India's latest surge, hopefully if a good T cell response was mounted to the vaccine, which we know that both AstraZeneca and Covaxin do, you will be protected against severe disease. And that really is a lot of data over the last, even before we had vaccines in the pandemic, showing how important the T cell response was to modulating severe disease. So if we look at the current vaccines then, if we look at the current vaccines all at once, the Moderna, Pfizer, Johnson & Johnson are the ones authorized in this country, but remember that those are, the top two are the mRNA vaccines. The third one is the adenovirus DNA vaccine. Then AstraZeneca, Bharat, and Sputnik 5 are the ones, Covishield and Covaxin are the AstraZeneca and Bharat. And they are both, the AstraZeneca is an adenovirus DNA vector. The Bharat is the inactivated whole virion, and Sputnik 5 is actually an adenovirus DNA as well. They all require 2 doses, except for the Johnson & Johnson, which is a 1 dose. And the fourth column is very important to show us that they all showed us that they not only produce neutralizing antibodies, but they produce strong CD4 cell protection with it, exactly the way you want it, Th1 geared response, including the Bharat a Covaxin product, which was that phase 1/2 trial data was published in the Lancet and showed a strong Th1 CD4 response in the phase 2 trial. The AstraZeneca also showed strong Th1 CD4 response and a CD8 response. And they actually in macaques showed that it protected against reinfection in macaques. The size of all of these trials was sizable. Bharat said that it was 22,000. Again, we don't have the full review. So this is the numbers in column 5 that are actually the numbers that got the vaccine. And all of them, including the updated data from Covaxin on April 21st in the press release, showed 100% protection against severe disease. They didn't define for us, again, the Covaxin trial was just the press release. We would want to see the full details, but they said that it was 100% protective against severe disease. There was actually one person in the Moderna trial who did get hospitalized for COVID. Otherwise, all of these trials showed 100% protection against severe disease. There were differences in the protection against mild disease, and the Bharat Pharmaceuticals one said 87%. AstraZeneca varies across the world, 76% protection in the US, 70% protection in the UK. And against the South Africa variant, it was actually halted. The trial was halted for mild infection, but it protected against severe disease. So we'll get into that just in a minute, a little bit more when we talk about these products. The 2 mRNA vaccines, the reason I want to tell you about them is I think they should be, well, I don't just think, they obviously should be donated to India of all this excess supply that's in the US. And I hope that donation will be soon. It should have been before this because there was a very large purchase of more supply than was needed for the US population. And we'll talk about waiving patents in a minute. Essentially, the Pfizer vaccine was 95% effective against symptomatic disease, and the Moderna was 94% protective. So they were very high against symptomatic disease, but importantly, they almost completely, as I told you before, protected against severe disease. Only the Moderna had one person that went into the hospital and then was fine, came back out. The Johnson & Johnson vaccine, which is the 1 dose, was also studied across a very large population, more diverse population because it was studied in Latin America and also Central and South Africa. So there was more diversity of participants, and there was more actually diversity in terms of the risk of severe disease. And there was a lot more symptomatic COVID-19 cases than any other setting. The Bharat trial has 127 cases. And again, all the hospitalizations and deaths were protected with the Johnson & Johnson vaccine. And it was also efficacious against the variants, but more variable against mild disease. So the Astrazeneca, which is Covishield that you have in India, at least locally produced, also has a very large trial design. And there was a strangeness about its trial design in the UK that people got half dose and then a full dose. But the Lancet publication, which is the more full set of data, did show that, that there was high vaccine efficacy. But importantly, again, with the severe outcomes, it was 100% protective, but its protection was 76%, at least in the UK study, and actually 76% in the US study and 70% in the UK study. Asymptomatic disease was also tested for in the Astrazeneca trial, and it reduced asymptomatic disease for about 60%. And importantly, and I think this is important because I know that India has adopted a strategy of waiting between doses, and that has caused anxiety because of the concern about how much circulating COVID-19 is going on. At least in the AstraZeneca trial, the longer between doses, the better it was for immunogenicity, meaning 12 weeks delay over 9 weeks, over 6 weeks was actually better. On the other hand, if you have a lot of, this trial was not done when they had the number of circulating cases that India has. And so I think India's strategy of 8 weeks is appropriate by this trial design. But the difference is that India, clearly people are being exposed at a higher rate than people are being exposed in the AstraZeneca trial. So the good and the bad is that it is cheaper and it's not as fragile as mRNA vaccines, and you can space out the dosing, but there are some rare clots associated with the AstraZeneca vaccine, a so-called vaccine-induced thrombotic thrombocytopenia, and it wasn't as protective against the B1351 against mild disease, though importantly, it was protective against severe disease. This is the Bharat phase 2 study that just showed us that CD4 cell responses, this is the Lancet study that showed us that CD4 responses were activated by the Covaxin vaccine, as were neutralizing antibodies. And that will speak to the variant question. This was actually the phase, the April 21st press release from Covaxin. And what they told us at least was that there were 127 symptomatic cases that were recorded in the phase 3 study of 22,000 participants. And that the vaccine efficacy was actually 78%. I misspoke when I said 87, but the efficacy against severe disease was 100%. So that's where we get this question of severe disease versus mild disease and the differences in those, which are really modulated by the antibody response versus the T cell response. Will vaccines work against variants? They do, but when there's so much circulating virus, again, it is against severe disease where your T cell response is most prominent. Even before vaccines came out, we knew that the T cell response to natural infection was very broad. Multiple T cells are formed against multiple parts of the virus, even multiple parts of the spike protein, which is what is actually mutated in the context of variants. So as a better example, because after vaccination, what is the response, the T cell response against variants? This is a study that was done with mRNA vaccination. And after mRNA vaccines, the blood basically of people who got mRNA vaccines were exposed to different variants. The UK, the B117, the B1351, the South Africa one, P1, which is the Brazil one, and the California variant. By the way, the B1617 in India combines a mutation from the South Africa variant and the California variant. And those are the two that what they mean when they said double mutant, even though there's actually more than one mutational across the spike protein. But luckily, with vaccination, T cell reactivity was preserved against all of these variants. And even in the AstraZeneca trial, and this is very important because again, they halted it. So many of us thought they should have continued that. In the AstraZeneca trial in South Africa, they showed that very robust responses were raised across the entire spike protein, where 87 different pieces of the spike protein, you had different CD4 and CD8 cells going to destroy each of those little pieces. And by having a variant, you knocked out only 12 of them, so that there were still 75 pieces of the spike protein that T cells could still respond to in the AstraZeneca vaccine. And then this is another study from the NIH that showed that across the spike protein, CD8 cells, different CD8 cells form across 52 little pieces, basically, of the spike protein, little pieces meaning epitopes or antigens. And so with the South Africa variant or the UK variant or the Brazil variant, only one of those epitopes, so-called epitopes, are knocked out. So that shows how in breadth and robust your T cell response is to vaccines. And then this was just a study from 2 days ago that the Qatar mass vaccination campaign that started in December 21st, and they recorded data out to the end of March, even after B117 and B1351 became the predominant variants, vaccine effectiveness, in this case, this was the Pfizer vaccine, was preserved against severe disease. So really shows the power of T cell immunity, even which is preserved against variants, at least to protect us against severe disease. And that's what made Covishield and Covaxin basically message that they are effective against the B1617, against the specific double mutation, even though more mild infections may break through. So this is why, though variants are of concern, I don't worry about them as much, because what I don't want people to get is sick. And essentially T cell responses do seem to be preserved against the variants and protect against severe disease. Now, do vaccines reduce transmission? And then I really, really want to get into your questions. I think it's important to say yes. And I don't, I think that we should never have had this narrative that vaccines wouldn't prevent transmission. In fact, I think that if vaccines had been given to India earlier, when they asked for it in October of 2020 in front of the World Trade Organization, we would be in a very different situation than we are now. Vaccines absolutely reduce transmission. They actually induce what's called IgA, which is an antibody that goes into your nose. They induce IgG, which goes into your nose. They induce T cell responses, and they also go into your nose. And we know that they induce IgA because we actually have good studies that show that. And all of that would prevent you from even getting asymptomatic infection, which you could pass on to others, even if you didn't know that you had the virus. And now there's study after study across healthcare workers in England, in Israel, across the general population in Israel. We have multiple studies in patients that present for pre-surgical procedures, for example, that vaccines will not only, of course, prevent symptomatic disease, like we've already spoken about, but they will prevent asymptomatic infection, which will lead to a reduction in transmission. Because if you don't know you have the infection and you could spread it, that's been the problem with SARS-CoV-2. So they absolutely block transmission. And before I get to the history of the patents, I do want to just tell us that the real world data on the vaccines have been very, very important, including among older people. And one thing I want to say, and then we'll get back to the patents, is that herd immunity, and the reason that it's so important to get populations vaccinated to have not been in the situation that we are in, is that herd immunity is essentially a form of indirect protection for others. So the more and the more that we get people vaccinated, or they have natural infection, which is of course not the way that we had wanted to get herd immunity, then we protect others around us, because our immunity will prevent the virus from spreading to, for example, children who can't get vaccinated. And there are places, of course, like the US, like the UK, like Israel, who are doing extremely well, and their cases are coming down very precipitously with mass vaccination. And that's why it gets back to my original point, which you were talking about at the beginning of this talk, which is that the most important thing we could have done, and I did write this article this week, and we didn't do right, was get at vaccine equity access. So to give you the history of this article is the important thing is that India actually appealed to the Trade Organization with South Africa in October and said, please waive the patents. And what do they mean by that? Well, in 1995, there was a stipulation, the World Trade Organization TRIPS agreement, that in the context of a medical emergency, that patents should be waived for life-saving medications or vaccines. And in fact, 1995, of course, was when that was actually written before antiretroviral therapy had been made available worldwide. In 1996, we had very effective antiretroviral therapies, but they were only affordable to rich countries, the US and Europe. And it was actually India, it was literally India that this is why we should have responded in October 2020, saved the world with antiretroviral therapy production. So in 2001, after 5 years of debating about with the World Trade Organization about waiving patents, India actually, Cipla Pharmaceuticals and others, made a cheap triple combination of antiretroviral therapy for the world. And they made it for $350 a year, which was less than $1 a day. South Africa purchased those medications from India, and the big pharmaceutical companies sued South Africa in 2001. And that was, they viewed very unfavorably by international pressure. And ultimately what was allowed to stand were these waiving of these patents from these life-saving medications from pharmaceutical companies, but it took years and years and years, and people dying of HIV needlessly in India and sub-Saharan Africa. And so for India to have appealed in October 2020, and first the US administration rejected it, then on March 5th, 2021, when Biden was in power, the big pharmaceutical companies wrote Biden in an open letter and said, we find this very unfortunate proposal by India and South Africa to waive patents. We don't think it will help. And as you know, just this week, the trade representative, Catherine Tai from the Biden administration, says that the US supports it, but the EU is not actually signing on to support this. So global vaccine equity is it's sitting here in the US, it's profound to me that people don't understand that we are, that just as things are feeling safer and better here, that like India's on fire and it's and Nepal, and it's terrible that we didn't respond to this in October. So I'll stop there actually and see because I know you said that there were a lot of questions, and I know we only have 23 more minutes, so I really want to get to your questions. Thank you.
Thank you so much, Dr. Gandhi, for such an interesting discussion. So we have some questions in the chat box right now. First question is by Dana Chu. So she asks, with the ongoing transmission, how worried should we be about variant of concerns and vaccine protection? I think will boosters be required? She touched upon that earlier, but like, will boosters be required? That's the main question.
Yeah. That's a great question. And I see the next question too. So I'm actually going to answer both of them by showing you slides as well. I mean, really what's going on right now in the region is not really because, I think that the variant B1617 could be more transmissible, is likely more transmissible, and that's contributing, but it isn't actually about evading the vaccines. And when we think about if boosters are going to be required, the important thing to remember when you think about the T cell response and the B cell response is that both of these types of cells make memory cells, and memory cells actually are kind of like putting in the bank cellular responses to vaccines. And then if they ever see the virus again, they are stimulated to produce new immune responses. So for example, let me explain that a little bit. T cell immunity is actually a very long-lived phenomenon. For example, there's a study that shows that measles vaccine can induce and keep T cells going for 34 years and counting. So your T cell response that you get from these vaccines will last a very long time. We know that T cells work against variants. We just discussed that. We know from the first SARS pandemic that I covered at the beginning, which was 2002, 2003, that 17 years later, people still have very strong active T cell immunity against the original SARS virus. And then importantly, there was a very nice study that showed that memory B cells were generated by these mRNA vaccines. They actually took people who got vaccine and biopsied their lymph nodes and saw very active B cells being produced, memory B cells in the germinal centers. Memory B cells last a very long time. In fact, there was a study from the 1918 pandemic, which I show up here on the top left of this slide, that showed that people who had been exposed to and gotten through the 1918 viral influenza pandemic 90 years later. So they took octogenarians and 90-year-olds, and they took them 90 years later and actually put their blood in a test tube with the 1918 influenza strain, and saw that they still had circulating memory B cells that could be stimulated to produce strong neutralizing antibody responses against the 1918 strain. So because of that, the fact that we form strong memory B cells and memory T cells to these vaccines and to natural infection, I'm actually not worried that we're going to need boosters. I think that's more of a pharmaceutical company money-making statement. So I'm not worried about boosters. And then I saw the next question, and I'll just answer that with the data, and then we can get to others. But it was a question on the children vaccine. The only Pfizer, actually the Moderna released their results today. I'm sorry, I don't have those on these slides. Those were from the age of 12 to 17. But the Pfizer 12 to 15 trial enrolled not that many participants, actually it was quite small, but of the participants who got the vaccine, who are 12 to 15, they did well, meaning they had a good safety signal. And also there was 100% efficacy against COVID-19 if you got the vaccine versus the placebo. There were only 18 cases, but they were all in the placebo arm. And I haven't reviewed the Moderna trial, which the press release just came out. So, so the EUA, at least for that, is expected next week here. I don't know if the CDC in this country is going to mandate that vaccine, because children are more spared the effects from SARS-CoV-2 than older individuals. But that's the data so far on the child vaccines.
Okay, thank you. All right, so next question is by Deepak Karki. Do you think a country like Nepal can use a second dose of Oxford vaccines manufactured other than Serum Institute of India, as there is a very thin chance of getting Covishield for next few months to Nepal?
I really do believe, yes, I would get them from anywhere. I mean, I actually really do believe that this is a very, very effective vaccine. And as long as it's, you know, anywhere creditable, I agree with you using it from anywhere. I'm positive the Serum Institute will be not distributing it elsewhere right now.
Okay, so next question is by Elena Pradhan. How important is technology transfer along with the IP waiver for India to start scaling up vaccine production, particularly J&J? If the IP is waived, what is the best estimate of India's vaccine production capacity? Are there any other South Asian countries with current capacity that could also ramp up vaccine production?
Yeah, I mean, that is a great question, because it is true that it can't just, I shouldn't have just said that it's just intellectual property, because it really is the intellectual property of the formula. It is technology, and it is also raw materials. And 2 weeks ago, I mean, actually just as late as just 14 days ago, there was a ban on the US had banned exports of raw materials to make these vaccines to India and other regions. And not this last weekend, but the weekend before that, export ban was luckily lifted. But it is more than this. And in terms of capacity, you would know better than I actually about capacity in other South Asian countries. The reason I'm so interested in IP transfer is for other countries besides the US and the EU to make vaccines, meaning anywhere that has the capability, if you have the IP waiver and you have the technology, anyone should be able to make these. Because even though they seem like they're really hard to make, it isn't actually that it's that hard. It's about some of the raw materials. It's about that lipid nanoparticle across the mRNA. To be honest, mRNA vaccines are not that hard to make because they just involve the sequence of the virus. They don't involve working with live virus in, like, for example, a biosafety lab. So they're not that hard to make. And that's why I think with the lipid nanoparticle technology and the raw material transfer can be made more readily anywhere.
Okay. But also, so continue with that conversation, that answer. But like mRNA vaccines, I think it's going to be because of the infrastructure in Nepal and India. Do you think they will be able to handle the ultra cold storage conditions required by Pfizer?
Yeah, I mean, so Pfizer is negative 13 degrees centigrade is the maximum temperature. And the Moderna one, and that's why many people have been urging the US government, because Moderna is still owned by the US government, actually can be at 30 degrees at -4 or between -2 and -8. And that is much easier to transport than the Pfizer. So I do think refrigeration is possible, but not the -13 is definitely harder.
Yeah. All right. So next question is by Lamo Sherpa. Thank you, Dr. Gandhi, all right. So I was wondering, for countries like Nepal that do not have the ability to produce and procure due to various reasons, its own vaccine and its ability to test what variants are driving the surge, what other precautionary measure do you see as appropriate at present to reduce transmission, especially in Nepal and in poor and densely populated districts that border with? What window of opportunity do we have in terms of time? So this is a common question going on in everyone's mind right now, especially in Nepal, as the cases are surging in an unprecedented way. So
Yes.
That's a great question. Yeah, we'd love to hear your thoughts about
No,
that.
it's a great question. And not unfortunately, there are tools. I mean, that's what we talked about at the beginning. It's just that it is obviously hard to have, you can't keep away from others when you live in a densely populated region. You can't, you can be outside as much as you can, which is better to be outside than inside. And because of the incredible, ventilation is natural outside. And then the mask is really important, and the quality of the mask is important. So even if you don't have some massive, very different, like an N95 that's mass produced, what you can do is take more commonly available surgical masks and combine that with a cloth mask when you're inside. Put those two together. And putting those two together is very effective for preventing transmission. So that's why I want to really stress that it's not just any mask inside. It may be any mask outside, but what's inside it really does require that fit and filtration of the double mask. Or if you have a single mask, then loop it as tightly as you can around your ears, double the cross over the loops and tuck it in on the sides.
Okay. And continuing with the mask conversation, so you've been affectionately known as the mask lady with all your papers and work on masks. What do you suggest? So you've also been a vociferous supporter of masking. Removing masking outdoors. So in this current situation in Nepal and India, where there's a lot of transmission, would you still recommend that?
It's a great question. Yeah. It's a great question. And actually, what we recommended in this article that we wrote just last week in the Wall Street Journal was in packed crowds and packed situations, even outside, I would wear a mask. So I would, for right now in India, it is very strange for me to be advocating for that in the United States and then talking about what's going on in India. And so it makes my Twitter feed look very confusing, I think, because there is this push towards normalcy in places that are vaccinating quickly. But in India, I would wear a basic mask outside in any crowded situation. And inside, I would wear that either double or fit it, tuck it in on the sides and double that loop. I really, really believe in the power of the mask inside. So outside, I would, in crowded populations in India, I'm not advocating for removal of the outside mask at this point, not with what's going on.
Okay. Thank you so much. And all right, next question is by Dr. Roshan. What is your opinion on the effects of steroids like prednisone on vaccines? Can such steroids be taken soon before or after vaccines?
No, it's a very good question, because if you're being treated, for example, for COVID with steroids, will this reduce the efficacy of the vaccines? It is true that steroids are immunosuppressant. They will block some of your immune responses. And so I think I have two suggestions. One is, if you can time steroids not around the time of the vaccine, if possible, at least 2 days before and at least 5 days after, I would wait. Because especially since we've seen the immune response rise very quickly, but I would want people to get at least 5 days without any steroids on board. And if steroids are mandatory when you get the vaccine, and again, we're sort of making this up as we go along, but I personally would think I would advocate for that person to get a booster, and yet another booster, like a third shot. If for some reason your vaccine effectiveness had to be brought down by needing steroids during the vaccine shot, just to give that profound effectiveness in the future, I would want that person to get a booster.
All right, thank you. And next question, it's a great question by Dr. Saurabh Wanta. So it's a question that's going in everyone's mind right now in Nepal. So because of the lack of vaccine availability, is there any possibility of combining the vaccines and for how long will the vaccine provide the protection? Could you highlight briefly? And along with that, and Dr. Binjwala Shrestha, who is from the same team, she wants to know if there's any update in mixing two different vaccines as a first and second dose, because we're limited by the number of vaccines that we will get. So some people have gotten the first dose of Covishield. They may not be getting the second dose for a while, So is it better to combine that with follow that up with Sinopharm, which is the other vaccine that's going on right now?
So actually, there was a study in mice that examined that the immune response is even better after mixing vaccines versus using the same vaccine for the second dose. This was actually specifically with an adenovirus DNA vaccine and then an mRNA vaccine. Has not been studied with the Sinovac, but I would say that there is no problem. They're actually studying that strategy now in the UK. There's an ongoing mix and match trial. There is no problem in my mind mixing vaccines. In fact, in a way, you could imagine that there could be an advantage of mixing one type of vaccine that would stimulate the immune response in one way, and then giving the second dose with another type of vaccine. So I see zero problem with that, and it could actually increase immunogenicity. And this speaks to actually a couple of these questions, because my incredible, when people say, what is your reach within the US? I don't actually have that much reach, but I want to reach, meaning I absolutely think that the US should be donating their excess vaccine supply, not just the Astrazeneca, which they said they would, which is 60 million doses. But we actually have a lot of excess doses of the mRNA vaccine as well. And so this is what when you ask at the end, if I would advocate for this, I will keep on advocating for this. And I think I know who to advocate with, but I don't have the direct line or anything I don't have the direct line, but I'm going to keep on advocating, because you're right, Nepal is severely lacking these resources. I did want to say one thing about the Sputnik though, because I see that as a question. The Sputnik vaccine is a good vaccine actually. And I didn't go over that, but that's an adenovirus DNA vector vaccine. It's 2 doses. It did show 91% efficacy against mild disease. And importantly, it did stimulate T cell responses with an interferon gamma assay in peripheral blood mononuclear cells. So actually, if there's a chance for Sputnik 5, I think that is a good vaccine. Okay, sorry, I kind of like read some of your questions, but please go ahead if there's any others.
Okay, so I think you've answered like most of the questions already from the chat box. All right, yes, going into the vaccine. So I don't know if you read the recent news about Brazil government like rejecting that vaccine because apparently they found some, even though it's supposed to be replication incompetent, they found some replication competent viral vectors in the batch that they got. So that's why they rejected it. Were you aware of that?
Yeah, I did see that. And I was hoping that was just that batch, because actually, when you look at the phase 1, 2, and phase 3 trial of the Sputnik 5 vaccine, they were very clear that they inactivated it so that it is not replication competent. So just like there were some problems with batches of Moderna, for example, in another way that caused more anaphylaxis here, I'm hoping that is just one specific problem, because the clear deactivation of the adenovirus is absolutely required for this. So when you're, I mean, someone brings up the Sinovac as well, that's a less effective vaccine. That is a less effective vaccine. But the important thing about any of these vaccines, in my mind, is that if there's a high prevalence in a region, if there is ongoing high cases like what you're experiencing, even a not as effective vaccine, which is what we thought all these vaccines were going to be, could bring down rates of infection. And then later we can think about getting a booster shot with another type of vaccine. But I would encourage Sinovac and Sputnik 5, both of these vaccines, in the context of high prevalence, because it will fundamentally bring down transmission.
So with the interest of time.
Yeah.
All right, thank you. So with the interest of time, so we just have 4 more minutes now. So one last question by Dr. Binjwala. So one of the frequently asked questions, all right, so how long will the vaccine provide the protection for? So especially this is in regards to anything else.
With that, I think I did sort of say that with the booster question, that with the raising of the memory B cell response and memory T cell response, I actually don't think, I think that vaccine protective immunity is going to last a long time. And I really do. So that my biggest goal, all of our biggest goals, is to get global vaccine equity, to get people vaccinated. But I don't think we're going to need, I think it's going to last a long time, because it's amazing that these studies actually showed us that they induced, by literally biopsying people's lymph nodes, that they induced memory B cells, which last, memory means 10 decades. Memory T cells also last a long time. So I think they will last a long time.
All right, thank you. So, all right, so we have 2 more minutes. So I just want to ask one last question to end the night. Why do you think, I just want to ask you, why were India and Nepal like spared during the first wave? So nothing really we did not see a lot of death. We did not see a lot of like cases spiking up, which made a lot of people believe that maybe we've triumphed over the SARS-CoV-2. A lot of experts in the area also believe that they're probably the population probably had some pre-existing immunity, which protected them from severe cases and death. And if I'm not mistaken, you also believe in the pre-existing immunity in that population.
I made a huge. I also made a huge mistake. I genuinely think I made a huge mistake. I also thought that, and I was wrong. I was completely wrong.
I also thought that too. I thought the same too, because it was just baffling that the cases were not rising up in that area, even though they should.
It was. So I completely agree with you I was mistaken, and I feel terrible about that, because it just seemed like it was going so well. And I think 2 things happened. Number one, we all overestimated immunity, like we did in Queens, for example. Like in New York City, there was a seroprevalence study in Queens that showed 60% seroprevalence, and everyone thought that New York City, when they went through their first wave, would have a lot of people with immunity, but that wasn't true. It was poor populations. And the same thing with the Lancet study in from July 2020 in the Mumbai poor urban slums. There was 60% seroprevalence, but only 11% seroprevalence in the general population. It meant that poor populations did get exposed, but not so many other populations that were able to stay away. And then when we release our guard, and when we had that much mixing that we had, and with the festivals and all that mixing that happened, then, and a B1617, which is likely more transmissible, I think it really took off. And so this is a mistake that, you know, I certainly made, and it feels terrible to have made that mistake. And I'm very sad for all what's happening there. And all I can think of is, all I can work on is getting vaccines there, because it is, you can use these other modalities. They should be used. The double mask is very important, but obviously immunity in a population is what gets us fundamentally through the pandemic.
All right. Thank you so much for such a delightful discussion, Dr. Gandhi. Thank you so much for making some time for us despite your hectic schedule. I know you had a lot of meetings back to back, so thank you so much for that. We learned a lot.
Thank you, and
we enjoyed
please,
this conversation.
yeah, please.
Hopefully, we can do this again with you in the
YouTube,
future.
Great, and send it to me, and I'll tweet it. Okay, thank you very much.
Definitely.
Thank you all.
Oh, definitely. Thank you so much, Dr.
Okay.
Gandhi.
Thank you. Okay, bye.